except that I'm seen as a shadow. I guess that's okay. So we're continuing through our chapter on the MHC, and I apologize for giving you so much last Wednesday. Kind of just got going, and I didn't see a place where I wanted to stop, and especially since I had allocated 3 lectures to it. But we got into a lot of stuff, and we got to present some real kind of classic immunology. Share my screen here. And we got into some really, when I think of just a few experiments to present in immunology. You know, those are some of the ones that we discussed last time, and we'll discuss today are some of the ones that we do that come to mind anyway. So reviewing a little bit, we had our, well, we go back to our picture of our MHC molecule with the fluor and I thought that was one of the slides that had could be divided in two. We see here the structure of the floor of the MHC and also the helices on the side, and highlighted their areas of high variability. And of course, where it's important, where peptides are bound. So different alleles can bind different peptides. So the polymorphism is functional. And you might imagine that, you can imagine, and it's certainly true, that there's great attention paid to those positions in peptides that will bind and won't bind, in looking at alleles that are associated with an autoimmune disease, for instance, type 1 diabetes, and those that aren't associated. So that's a big area. And of course, the short answer is, the answer is that the answer doesn't jump out at one. Okay, here's the clear reason why a certain MHC allele is associated with diabetes and another one is not. It's, you know, we haven't solved it, so not easy to find. So we talked about MHC class 1 and class 2, similar in structure, but of course different. MHC class 1, mostly presenting intracellular peptides. We'll discuss that further, and exceptions there. So the idea is checking that cells are safe and are okay. So you can imagine the T cells going along and interrogating all cells and saying, you're okay, no, we have an invader here. We need to act. And if there has been an infection with a virus, so we say class 1 expressing endogenous peptides, so that means yes, those good peptides made by the cell itself, but also endogenous, meaning produced on the cell's own ribosomes, perhaps by genetic material from an invader such as a virus. And in general, MHC class 2, extracellular, so debris, a free protein perhaps. And these in general activate helper T cells, of course. MHC class 1 on essentially all cells, all nucleated cells, and class 2 on the professional antigen presenting cells. Dendritic cells, macrophages, and B cells. And, these guys are non-professional. MHC expression can be upregulated for certain conditions. Viruses make sense that viruses will not try in a conscious sense, of course, but evolution has led to the viruses having a good adaptation to try to stop class 1 expression. A number of viruses make a protein that'll bind class 1 and prevent it from going to the cell surface. MHC alleles, first known as immune responsiveness or IR genes. How is this? Is this because some alleles can bind a key peptide and others can't, or is there a hole in the repertoire with T cells with different abilities to have TCRs interact, and both have some validity. So then we get on to our discussion of MHC restriction. Key experiment, we gave two from the book. I'll review that again. On the right, the Zinkernagel and Doherty. You know, Zinkernagel is one of those names you don't forget, right? You know, it's not like, you know, if it was Smith, the experiment done by Smith and Jones, you know, we'd forget about it. Zinkernagel, yeah, okay. Anyway, it's demonstrating MHC restriction. Remember when this was done? This is before people knew that there was a what we now know as the T cell receptor. So this was quite impressive for that time. And we observed that the killing cells, the T subscript C, and the presenting cells need to be of the same haplotype. And of course, later it's understood how that develops. And so this mouse infected with LCMV, lymphochoriomeningitis virus, has a history, a big history in basic science, basic immunology. I'm not sure how much of a concern it is health-wise. It seems to me, since I've been aware of it, it seems to me I've heard of something related to people with this virus. Note one thing with a viral response, you might, at least some cases, viruses can give a very strong response compared to some other antigens. So idea here is the mouse of a particular haplotype is infected. Sometime later, obtaining spleen cells. It's a classic kind of experiment with the mouse. And then incubating those spleen cells with various potential target cells. The assay here, notice it says chromium-51. Historically, this has been the widest type of assay for killing used. In the target cells are loaded with chromium-51 through osmotic shock. They take up that chromium-51, and then if the cells are lysed, that chromium goes into the media. So a way to determine killing. And control target cells that are not infected, no killing, not a surprise. Because these T cells, these killer T cells were stimulated with the virus. The experiment in the middle with the target cells from the same strain of mice that were that were infected, they are killed, leading to release of chromium-51. Well, not a surprise, I guess, especially given what we know now. But the key experiment that maybe they had some inkling from the guinea pig experiment, but here on the far right, when the target cells were infected with the virus, the LCMV, but they were of a different haplotype. Okay. So they were B target cells, no killing. So the response was restricted to the right haplotype. Okay. So again, it's important to remember this, and the way this has worked out, this MHC restriction. And recall now, so now we know the explanation for that. We now know that there is something called the T cell receptor, and we recognize the ligand is peptide presented by MHC. So we recognize that the T cell receptor recognizes this compound ligand, that one molecule on the T cell recognizes this ligand. And that, of course, all makes sense. This is before people really knew that there was such a thing as a T cell receptor, let alone that it recognized two things at the same time. Could have been recognizing separate places on the surface. Okay? So a very important thing here in our development. And a slightly different experiment here with the guinea pigs and working with T cell proliferation. Just as an aside, historically, I guess it's still we typically measure activation of helper T cells by their proliferation. When they get the right stimulus, they divide. Sometimes people measure incorporation of nucleotides, or could just measure total numbers of cells. Those are both widely used. And then for CTLs, a killing acid, and usually with chromium. So historically, that's how that has been done. So now I went on and I presented this in the asynchronous lecture Friday. Antigen processing is required for T helper cell activation, and again, a fundamental thing about immunology. So we talk about these areas, title antigen processing or antigen presentation. How does this actually happen? So we talked about restriction earlier, but then it was discovered that whole antigens cannot be recognized by T cells. This is a surprise because remember, with B cells and the B cell receptor, they can recognize whole antigen, and sometimes a three-dimensional structure, a three-dimensional part of the antigen would be recognized. So this is the kind of experiment that was done. I don't know if any of you in any of your various lab endeavors ever done fixation of cells using yes. So typically aldehydes, formaldehyde, glutaraldehyde, basically crosslinks the hell out of cells. So many crosslinks form, covalent linkages. So the cell, everything is still there, but it can't do anything. Sometimes fixation is done when you don't want one cell type in a mix to be able to divide and others can. Anyway, in this case, it's used to stop some cellular processes, and the experiment is done in this way, that if an antigen presenting cell is first fixed, as in the upper figure here, first fixed, and then antigen is applied, that cell cannot present to a helper T cell. And our interpretation is because something's going on that was blocked by fixation. Okay? Think of the cell being frozen. Okay? Can't do anything. And here in the middle figure here, if the antigen is given and then allowed 1 hour of time is allowed to elapse, something's going on, which we now have a handle on, and then fixation, now it can present to a helper T cell. So there is T cell activation. And an interesting experiment here, of course, all these experiments, one wonders how much they kind of thought they knew from some preliminary experiments. But here, when they do the fixation first, and then add antigenic peptides, not the whole protein, now presentation does occur. Okay. So this suggests that what's going on in the cell is processing whole proteins being broken down to peptides. We now know the size of peptides that are presented by MHC molecules, and we count. And now we believe we understand this. That's an important point also experimentally, that people can add peptides to antigen-presenting cells and see the response. Okay, so you can test, does this peptide work or does this peptide work? You can have a panel of peptides that you can make in the lab on a peptide synthesizer, or you could make a set of peptides that are variants on one natural one to see what if you substitute this peptide with this amino acid, this position. So it's easy to do that, synthesize those peptides, add them to antigen-presenting cells in an assay, and then add the T cells. Okay. So experimentally, that's how that can be done. But also, this brings up this point and begs question, just what's going on with this antigen processing? What enzymes do it? How do they do it? So this is where the cell biologists come in, and people who are very knowledgeable about cell biology. You might recall that from your class. The kind of thing they do is look at different organelles within a cell, identify them sometimes by their density, or be able to see them because a certain particle will accumulate there. And now these guys come into play in looking at the pathways for breaking down protein. Yes?
Are the cells that are fixed dead?
Are the cells that are fixed dead?
Yeah, are the cells
They won't divide.
So they are not dead yet.
But they can still, some things they can do, yeah. Okay. So people began looking at processing and class 1 and class 2 differences. And remember now, we've already talked about most of the peptides found on the surface on class 1 were endogenous, and those on class 2 were exogenous. Now they do their processing, and this figure describes that.
Question.
Yeah.
What's happening in the Golgi? Is it just because they're already in a vesicle, right?
So sugars get put on the Golgi. Might be some proteolytic processing as well. So some key experiments done here. Bracciali has mentioned some important experiments. Found that class 1 presentation required protein synthesis using needed to use a replication competent virus. Class 2 did not, but class 2 was blocked by chloroquine. Chloroquine has classically been used to block some stages in endosome processing. Okay, so people really get into this: early stage endosomes, late stage endosomes. Compartments become more acidic and further along, and different proteases are then active. Okay, so indication that these pathways were different. So the endogenous pathway makes use of proteasomes. So this is something I don't know, I think we often gloss over. Regular cells have proteasomes, but the antigen-presenting cells have special proteasomes. And here it says there are some peptides generated by both, and some are specific to the immunoproteasome. So these are produced peptides are the right size for MHC molecules. TAP genes are found to be necessary, standing for transporter associated with antigen processing. Mutants were found called RMAS cells. The name associated with that was Townsend, and he found that peptides were necessary to stabilize the beta-2 microglobulin class 1 interaction, and defect was in transport of the peptides, and it was in these TAP genes. So transport from the cytosol to the rough ER where class 1 is made. ATP is required for this. TAP also, the genes are also in the MHC region. TAP has some specificity for the right size peptides and with an anchor in place. So that was an important advance in antigen processing. Chaperones, proteins that bind to proteins, to other proteins, and they're made that prevent them from aggregating. Talked about, and some were found to be to aid peptide assembly with MHC class 1 molecules. And some of these were found important, and including peptidases that would trim to the right size for MHC class 1. We see here the cytosol and the RER, rough endoplasmic reticulum lumen, class 1, aided by a chaperone. Yeah.
Do you know, or do we know why the class 1 and class 2 have like different lengths that they chop to?
You mean why is it functional, or how they actually?
Yeah, is there a function, or is that just something that happened evolutionarily along the way?
Yeah, I mean, I think as far as why it's beneficial, or I think the enzyme talked about trims this, but as far as why it's beneficial to do it this way, I can wave my arms and say maybe the killing is more important. That you'd want to limit to maybe a little more play with the class 2. I don't know, but that's really an arm waving kind of thing. So associating with beta-2 microglobulin, and TAP provides helps provide peptides.
I have a question.
Yeah.
What is the function of the beta-2 microglobulin?
It's necessary for the final structure of class 1. It doesn't come to the surface without it. It is not polymorphic. So that's kind of interesting, whereas everything else is. A general rule for these complexes is that they don't come to this, one protein doesn't come to the surface without the others. And that's the case there. But there's no, it's not polymorphic. In fact, you can infect, you can infect human cells the other way around. I know you can do mouse, I've done this with mouse cells with, you can put in a mouse class 1 with into human cells, and it'll associate with the endogenous beta-2 microglobulin. Anyway, yeah.
Is that the same for class 1 and class 2?
Well, no, because in class 2, the two different, the alpha and the beta are both polymorphic.
So there's redundancies in that.
Well, I mean, when we use the word redundancy, it's just more that's part of the variability.
Okay.
So, yeah, but that is an important difference, that all the polymorphisms are in both alpha and beta in class 2.
Well, no, I was asking about, sorry, I wasn't clear, about the,
Do they come to the surface?
the fact that to come to the surface, they're all needed, because it seems
Oh,
like
yes,
that would
yes,
make
that
it
is true.
really
Yes,
vulnerable
yes,
to
yes,
evasion.
Yes, yeah. I think just the folding is off. You know.
Okay.
And just as another thing that comes to mind, like CD3 for TCRs need all the CD3 and they all come together. And I think if they're missing any one thing, that they don't come to the surface. Sorry for confusion there.
Oh, that was on me.
So chaperones assist with class 1 maturation. The end, class 1 exits and goes to the Golgi. The exogenous pathway is a somewhat different story. So the antigens are internalized, they go into endosomes, and there's a whole thing about endosomes fusing with lysosomes, becoming those early and late endosomes, and I think the later ones are always more acidic. And there's a lot of effort into looking at pathways here, the order we see them. So antigens taken up, go into increasingly acidic compartments, early and late endosomes, endolysosomes, and lysosomes. Loading occurs in the M2C, a unique compartment. People use chloroquine. You heard about people, the hydroxychloroquine for fighting, cell biologists have been using chloroquine for a long time. People use chloroquine to increase the pH and disrupt this degradation and block this. People also try using protease inhibitors to disrupt. So a couple tactics here. Protease inhibitors to block a certain class. So remember, proteases are needed for the antigen processing, but also note that a protease that cuts in the middle of an epitope would also block the presentation of that epitope. So there's a lot of stuff there with proteases. So people do things like work with protease inhibitors, and even quite a few years ago, we're making knockouts of some of the proteases to disrupt this whole thing. Okay. MHC class 2 is produced. It's associated with something called invariant chain. That's capital I and little I. So it's associated with new class 2, presumably because this groove is attractive or something to fit in there, and presumably to not allow endogenous peptides to now come in. Okay? And the invariant chain has a signal for sorting from the Golgi to the endocytic compartments. It's a bit of a complex slide here. So we see MHC class 2 bound to invariant chain. Invariant chain is trimmed, and the remaining bit is called clip, which is associated there, and eventually the antigenic peptide replaces it. So peptides displace clip. There's another molecule encoded in the HLA region called DM. It has an editing function, removing clip and others. Class 2 and peptide eventually goes to the surface, it becomes stable. DM was discovered because there were a genetic approach, there were mutants that were found that had some defects in antigen presentation. Key factors here: invariant chain, forms binding early on, but then allows itself to be displaced, allows sorting, degraded by proteolytic activity, clip remains, and then DM is involved in exchanging clip. People study this to the nth degree, okay, to find everything about all this processing.
Really quick.
Yeah.
HLA-DO modulates the function of HLA-DM. What does that mean? What is it modulating?
You know, I think that's kind of an arm-waving thing, and you know, I really know a bit about DM, and they found DM mutants didn't present well, but I think in vitro there's some evidence that DO can act antagonistically with DM, but really, yeah, I couldn't really give you a good answer on that. So this is return to our earlier slide with more detail. So class 1, endogenous antigen degraded by the proteasome, peptide transported to the ER by TAP. Binding chaperones. And you see captures peptide. Now it's transported to the surface. Let's look at class 2. Invariant chain binds class 2 early on, blocking other things from binding, especially endogenous. And you see class 2 is routed to endocytic compartments. Invariant chain is degraded, leaving one fragment clip. Exogenous antigen is degraded and eventually replaces clip. And DM mediates this exchange. Now it goes to the membrane. Okay, a lot of discussion of that. Okay, yeah, the next section is titled Unconventional Antigen Processing and Presentation. So, like everything else in immunology, in biology, and especially immunology, there's exceptions to rules. So, in one of those exceptions is that exogenous antigen can sometimes be processed and presented by class 1, and sometimes endogenous by class 2. Okay. So this helps in the immune response. So this involves, it's called cross-presentation, and cross-priming are used here. So it's been observed, there's a little bit of a controversy here. In vitro, they found some evidence that other antigen-presenting cells could do this, but in vivo, I believe they could only observe that dendritic cells could do this cross-presentation. Yes?
Is that because it's mistaken for something that's exogenous or endogenous, or because it, again, is like a redundancy built into the system?
I think it has value. No, it definitely has value
okay.
for the immune system. So in that sense, it's not a mistake.
Yeah, I think the scenario given by the book is how do you get
do you get, what?
How do you get cytotoxic T cells to kill? I'm sorry, there's a scenario in the book. I cannot.
Yeah, but it's something, no, it's beneficial in that something that would only be observed an exogenous antigen. If it was only going into class 2, then you would not be able to get CTLs to kill them. So there's clearly a benefit. So people have wondered, how does this happen? Do only some dendritic cells do this and others not? So people have kind of, I think this is a little bit of an arm waving thing, but they've suggested that in order to do this, that dendritic cells may have to be licensed. So here we have the idea of a dendritic cell being interacting with a T helper cell and stimulating that T helper cell, but also that T helper cell also sending signals back to the dendritic cell. So now it's licensed to redirect exogenous antigen into the endogenous pathway. So there's some evidence for this. So back and forth signaling going both ways. By the way, the guy, perhaps the most prominent person in this cross presentation has been the guy I've mentioned a lot, who's retired now, but the most prominent immunologist at the U a few years ago was Mike Bevan. So here it says it kind of avoids that controversy, saying dendritic cells appear to be the primary cross-presenting cell type. Again, exogenous directed towards the endogenous pathway, and this allows for presentation on class 1, priming killer T cell responses. And it does say here, the only ABC so far to exhibit this activity in vivo. So now we have that dendritic cell, perhaps because if it's been licensed, now it's capable of killing. So cross presentation and cross priming. Okay. So what should be going to the endogenous pathway going to the exogenous or vice versa. Okay. Lastly, I think this is one of the sections that I said don't worry about, but I will mention this to you. So sometimes there are non-protein antigens recognized by T cells, and these are presented by so-called non-classical MHC molecules. CD1, so this is a CD1 family of non-classical class 1. They present non-protein antigens like mycolic acid. Mycolic acid, remember from micro, is a really, really long chain, very hydrophobic acid from Mycobacterium, the tuberculosis organism. So, and some other lipids. So these genes are non-classical, they're not in the MHC locus. Several CD1 type genes are known. Little polymorphism, somewhat similar to class 2. We see here, supposed to get out of this is there's a deep groove in CD1 at lipid antigen binding. The lipid going into the antigen binding groove. Okay, summing up, T cells, need peptides presented to them for activation, understanding them. It's crucial to understanding T cell activation and how they're processed. So yeah, processing is very important, and things still aren't completely worked out as far as different cell types and proteases for processing. And remember, on the one hand, degradation is necessary to cut up, but also too much degradation, or say cutting in the middle of an epitope that would be recognized by a T cell, that would be harmful, right? So kind of dual, two sides of a double sword, two edges of the same sword. Yeah. Okay. So these are, of course, areas under study, and people try to perturb the system with inhibitors of proteases, following antigens through different compartments, knockouts of protease genes, those kinds of approaches. And of course, much effort into looking at what kind of can all dendritic cells do this. I think this is one thing where it's been suggested that the lymphoid-derived dendritic cells can do cross-presentation, but the myeloid cannot, I believe. That was suggested at one time, but not simple experiments to do. Okay, I think I will leave it at that. And yeah, sorry for not dividing things up well. I could have saved more for today and done less last Wednesday. But big thing on the MHC, interesting in genetics for the MHC and all this stuff that's involved. It's a really remarkable book, because you can see why there's a whole field called immunogenetics, right? And this is where the imaging processing comes in. There was the institute where I worked, there was a woman whose expertise was mainly the cell biology, so was really into this, the fine points of this, isolating slightly different compartments. And of course, sometimes you do this with tracer antigens, things you can follow. Of course, then you worry about, are you perturbing the system by different things you're adding, especially if it's something you can follow because it has say a heavy atom on it or something like that. Anyway, so a big area for antigen processing and presentation on the surface. The fixation experiments are certainly very, very important, and also the result that one can feed peptides and do experiments with them, because this allows one to either chop up in the lab or make synthetic peptides. Say if you have a 100 base long protein, you can do things like make peptide 1 to 10, 5 to 15, 10 to 20, staggered across the whole length of the peptide, and feed them and see which of them will respond, will stimulate T cells, and get an idea about which ones work. Okay, very good. Any other questions about anything? Okay, I will stop my share there and stop the recording.
Actually, I have a question. It's about nomenclature with the MHC 1, 2, and 3. If MHC3 is different in both form and function, why is it still in this series of 1, 2, and 3?
Good. I really don't know.
Okay.
Yeah. And I don't know. I've never heard that discussed. I don't think there's. I think there's still decreased recombination there, but I don't know about that. Yeah, there's a lot of stuff to unclear.